Hello everybody, thank you for taking the time to join today's webinar. I'm excited to talk to you today about the Sunny Suite and how it can help you in your development of next level LNPs. Let's get straight into it. Genetic therapies are behind some of the most groundbreaking therapeutic developments in recent years and have been shown the potential to be the basis of many next generation medicines. Gene therapies, typically delivered by some form of therapeutic nanoparticle, have been used in the treatment of a number of ocular disorders such as corneal, choroidal, and retinal diseases, improving efficacy and reducing immune response. In ongoing clinical trials, a potential gene therapy treatment for cystic fibrosis is being tested, in which a corrected version of the CFTR gene would be delivered into the patient's cells. Gene editing has also shown promise in correcting the genomic defect that causes sickle cell disease and leukemia, as well as restoring hearing in deaf children suffering from genetic auditory neuropathy. So there are a number of fantastic applications for gene therapy already demonstrated, and I'm sure many, many more to come. Most of these types of therapies involve the delivery of RNA into the cell cytoplasm, or DNA into the nucleus. Messenger RNA-based therapies target the production of specific proteins. Now, this could be instructing the body to make a missing protein or a protein that can trigger a desirable immune response, as in the case of an mRNA vaccine. mRNA tends to be relatively short-lived in the cell, and so repeat doses can be required to maintain a therapeutic effect. Other RNA types include antisense oligos, which can silence genes, and small interfering RNA, which targets a specific mRNA in the cell to prevent it from producing an unwanted protein. This is how the clinically approved drug Onpattro works to treat hereditary transthyretin-mediated amyloidosis. DNA therapeutics require delivery into the nucleus of the cell, where the synthetic DNA is either inserted into existing DNA or sits alongside it. DNA contains the coded instructions for the cell to produce a specific RNA or protein. There are multiple approved therapies for viral vector-based delivery of DNA. However, these are typically limited to a single-dose treatment due to the triggered immune response by the viral vector. As an alternative to viral delivery methods, LNPs are a highly promising non-viral delivery vector that overcome limitations such as restricted cargo capacity, manufacturing complexity, as well as some safety concerns. LNPs are kind of like Amazon boxes. The same components, internal and external packaging materials in the form of different lipid mixtures, can be used to safely package and deliver a range of different cargo types and sizes to a specific delivery site. By varying the composition of the LNP, you can tune the particle to suit different cargo types, adjust the targeting behavior, a bit like an address label on the box, and the particle size can vary to accommodate different sizes of cargo. A prime editor DNA at 8 to 10,000 base pairs is much larger than a simple mRNA construct for which viral vectors might be suitable. For the larger prime editor DNAs, the viral vector isn't large enough to contain that construct. There are a range of ways that LNPs have historically been made, but solvent injection is now the prevailing method of producing these particles, whether at an R&D stage of development or in manufacturing. Solvent injection refers to the process of mixing a stream of solvent with a stream of aqueous buffer in order to trigger the self-assembly of nanoparticles as the polarity of the mixing streams changes. These lipids are typically dissolved in a solvent such as ethanol, and the cargo, be it RNA or DNA, is usually dissolved in an acidic buffer such as citrate or acetate. As the solvent mixes with the acidic buffer, the RNA begins to condense and charged lipids begin binding to the RNA, forming a complex. As mixing continues, these complexes are surrounded by the other lipids in the solution and consolidate to form a lipid nanoparticle, with the pegylated lipids on the outer surface increasing stability in solution. Researchers developing novel genetic medicines undergo a lengthy development process in order to eventually reach clinical trials and production. During early stage R&D, the ideal therapeutic nucleic acid must first be developed, which can involve screening a large number of different constructs for signs of promising efficacy in vitro or in vivo. The same is then true for screening the formulation of the lipid nanoparticle to encapsulate and deliver the RNA, and to improve targeting and residence time within the body, for example. Once the construct and the LNP formulation are finalized, the next step is to optimize the process parameters such as the mixing chip use, the inline dilution, and to ensure that the process is suitable for larger volume manufacturing. So once you have the formulation and the process pinned down, you can move into clinical trials, which involve injecting LNPs into a population of humans, increasing numbers of people through the different trials towards commercial release of the drug. And so that involves continuous regulated particle production in a GMP environment. This entire process covers multiple labs, different objectives, and therefore different requirements for equipment at each development stage. The whole process can be time and material intensive, involve significant equipment investments, and complex process transfer between those different bits of equipment if they're based on different process technologies. So let's talk briefly about what's needed at the different stages of development. As researchers move through the different stages of creating new nanomedicines, the needs of the types of researchers will vary in terms of the number of formulations that they want to change, the types of process parameters they're looking to investigate, and also the scale of production that's needed. So at early discovery stage, it might be that you're exploring many different formulations in a large number of experiments with relatively fixed process parameters, whilst wanting to keep production volumes low. In this instance, a format like a well plate is ideal, where we have small combined volumes in large numbers of different formulations. As we move towards development and optimization, we may want to start varying more process parameters and exploring the best way to form the particular particle formulation that we've created. That process typically might involve producing some slightly larger volumes due to a need to do more detailed and involved downstream characterization and efficacy studies. So for this, we might look to a system which can still automate experiments, but collect a number of different samples. And then as we move to production, we're going to start thinking about a fixed formulation, fixed process, and continuous production into larger collection vessels in order to undertake clinical trials and ultimately to commercialize our drug. Another critical consideration in any process being developed in R&D is to keep an eye on the end goal of manufacturing and the feasibility of process transfer. For many commercial solutions, transitioning from early stage R&D through to preclinical development and finally to GMP manufacturing, a different pumping method and mixer type can be used between different instruments, and that means a constant re-optimization and revalidation of your particle production at every stage. But why not keep things simple by using a platform with consistent pumping and mixing technology all the way from screening to GMP? That's where the SunnySuite comes in. With the SunnySuite products, Sunscreen, Sunshine, and Sunbather, you can produce LNPs from a few hundred microliters on a 96 well plate format up to liters of product using the same pumping technology and mixer designs. And at every stage of development, Stunner can be used to get rapid, detailed characterization of your LNP and its precious cargo with 2-in-1 DLS and UV-Vis. In the rest of this presentation, we're going to take a quick look at some of the promising developments we've seen published in just the past few months that highlight just how impactful the first step of formulation screening can be on advancing LNP therapies. Then I'll share with you how all of the SunnySuite products can help researchers accelerate their formulation development and transition into manufacturing. And at the end of the presentation, I'll give some examples of how our users have benefited from these tools in practice. So let's talk briefly about formulations. LNP formulation development is a research area rich with opportunity to improve the quality, targeting, efficacy, and overall performance of nucleic acid drug delivery. But with so many variables to explore, just like our prospector here, it can often feel like it takes a lot of sifting to hit upon the most promising formulations. as well as being able to see a route to manufacturing. The days in which all LNPs are formed from the same basic blueprint are numbered. The standard formulation of ionizable lipid, structural lipid, cholesterol, and PEG lipid at a 50:10 to 38.5 to 1.5 ratio is being replaced by a multitude of different formulation paradigms. So let's take a look at how each of those individual lipid components is being explored in order to progress the field of RNA LNP drug delivery. Let's start with the structural lipids. Increasing the proportion of structural lipid and adjusting the ratios of the other components in the LNP has led to a number of interesting developments recently. Just as a couple of examples, in this paper from the Peer Lab, increasing DSPC content in certain formulations actually led to higher efficiencies in targeting the colon. And the Cullis group, exploring a similar variable, increasing the amount of DSPC, created long circulating LNPs to improve the residence time within the body, and found that also by exchanging the DSPC for sphingomyelin showed even more efficacy, leading to excellent retention time in vivo and high transfection efficiencies too. The search for alternatives to the hydrophilic stabilizing PEG component of LNPs is also being explored in detail. The two papers shown here examine different techniques for improving the PEG performance. On the left, its publication shows grafting short PEG moieties, which are too short for immune recognition, onto a polyamide backbone, which ensures improved delivery when we have repeat doses to prevent the immune response of the body from blocking and degrading the LNP once the PEG has been recognized by the immune system. And on the right-hand side, this shows synthesizing a PEG alternative via raft polymerization, which has demonstrated to improve in vivo luciferase expression relative to standard PEG constructs. Understandably, extrahepatic targeting of LNPs is one of the biggest areas of research going on at the moment. The technologies available to target LNPs to different specific tissue types or cell types using targeting elements rather than lipid components is also developing rapidly. These range from examples like as shown on the left here, using small computationally generated peptide fragments to attack targets behind the blood-brain barrier, to using bispecific antibody fragments which can easily couple various targeting elements to the LNP surface with minimal chemical modification. Obviously, for the field of LNP therapies, being able to target effectively to the correct organs and tissue types is one of the biggest leaps forward we can take in this field in the near future. And whilst by far and away the most common formulation combination involves those typical 4 lipid components I described earlier, both 3 and 5 lipid component LNPs have been making an appearance in recent years, with effective lung targeting realized by removing the cholesterol component, as well as exchanging the neutral DSPC lipid for cationic DOTAP, which really reveals the potential flexibility of the lipid formulation on the particle structure. On the other hand, the addition of components has also made an appearance, with cholesterol analogues and other small molecules playing a role in enhancing nucleic acid delivery and targeting. And last but not least, of course, there have been a wide range of developments in the structures of ionizable lipids. Examples include the use of branch-tail biodegradable lipids being developed to improve potency, making endosomal escape more efficient, or using dendron-like lipids to improve the targeting of the spleen, or changing the structure of the hydrophobic portion of the lipid to influence the particle biodistribution in mice in favor of the spleen or the liver, depending on the tail length of the lipid, not the mouse. With all of these exciting developments involving formulation adjustments, the method involves running a whole bunch of LNP synthesis experiments and using particle characterization tools to identify the signs of gold amongst the sand. And a lot of formulations can end up making sand. Sunscreen lets you produce sequential automated LNP formulations at a rate of one experiment every 3 to 4 minutes, amounting to a total of around 5 to 6 hours for a full 96 well plate, which allows you to vary the RNA cargo being encapsulated and the lipid formulations being used. This means Sunscreen can be used to boost your RNA development workflow, screening different RNA constructs for efficacy using a baseline LNP formulation, as well as when you're then searching for that killer formulation to perfect the delivery of your RNA. The system also has inline dilution and user control over things like washing levels and collection conditions like the head and tail cut and the total volumes collected. And these can be varied between each individual experiment in your table of experiments. The typical low volume formulation is around 400 microliters at a 3:1 ratio, and the instrument mixes together reagents on the Sunny. These are glass reusable microfluidic mixers which get automatically cleaned between each experiment, along with the rest of the flow path. These low volume experiments are run by releasing reagent into a continuously flowing stream, making our experiment directly translatable to our continuous flow LNP instruments, Sunshine and Sunbather. And with a size variance of less than 1% across an entire plate of replicate formulations, you can be confident in the integrity of your particle synthesis with Sunscreen. The Sunnies come in a range of geometries and sizes to suit your application needs, and are precision manufactured to ensure incredibly consistent channel dimensions and super smooth contact surfaces. Sunnies all have consistent positions for fluid inputs and outputs, so swapping from one Sunny to another is a breeze. It takes around 15 to 20 seconds at the instrument. Our Sunny Trident T, for example, is excellent for screening with its larger channel widths, optional inline dilution built onto the same Sunny, and a great tunability of particle size. If you prefer a more traditional T-mixing approach, we have you covered there as well. We've got 3 different sizes of T-mixer and also 3 different sizes of X-mixer as well. And lastly, we do have micro-mixer chip geometries and a 3D pour geometry as well. Setting up experiments in Sunscreen really is a breeze. Once you've decided on a Sunny and a set of conditions, you can design your experiment. The Sunny Suite software allows you to do that using an imported Excel spreadsheet, which automatically validates your experiments as you type them in, making it easy to plan your experiments from anywhere. Here we've set a sample size of 800 microliters collection with a 10% head and tail cut being discarded to waste, but this can be customized to your testing needs. Sunscreen will even tell you the runtime and the estimated driving fluid usage so that you can plan around the system's run. It'll give you a schematic to help guide you in adding material to your well plates so you can see how much of each reagent you need to add into each well, and the same for collection. We often design full 96 experiment runs, but we have customers that do all sorts, from full plates to half plates or even just a row at a time, giving you time to process and analyze one set of experiments while you move on to the next. Here you can see an experiment running on Sunscreen with colored dyes. The flow path before and after the sample is filled with driving fluids, which is typically the solvents in which your reagent is dissolved, so ethanol for the lipids and acidic buffer for the RNA. Your precious reagents are inserted into the flow path and carefully collected using a series of sample loops and valves, which minimize the amount of reagent consumed in each experiment. When you're screening hundreds of experiments, you want to be confident in the consistency of the process you're using. With Sunscreen, you can produce highly consistent data from repeat experiments, even if you just ran a not so good formulation directly before it. The data you can see on the screen here shows 90 identical repeat experiments using a DOTAP lipid formulation. And we've included 3 sets of experiments duplicated, which are the red bars you can see, which left out the PEG from the formulation. So these represent poor formulations that caused aggregation and large particle sizes and PDIs. you can see that the data for the repeated experiments of good quality LMPs are completely unaffected by the presence of the bad samples, and the CV of those 90 repeats is less than 1%. So you can be sure when you run a plate of formulations on sunscreen, you can be confident in your data. Between each individual experiment, the system is carrying out an automated wash and clean between experiments, and your wash volume is user adjustable. So you don't have to worry about a bad formulation affecting your potential hit. And it means that when you are doing a full screen, you can interpret that data knowing that your underlying process is not a variable from run to run. Knowing that we have confidence in our process, the screening data here shows just how quickly you can gain insights on your formulation changes. With these 32 experiments run in triplicate and characterized on Stunner within a single day, with only around an hour of hands-on time at the instruments themselves. In the screen, we varied the cationic lipid, the structural lipid, the PEG lipid, and the PEG mole percent. You can see at a glance there's a repeating trend of decreasing size as the mole percent of PEG increases. Additionally, we can identify more nuanced trends, such as the combination of reagents that lead to trends like lower size or PDI, and also to identify formulation combinations to watch out for that give poorer encapsulation efficiency or more particle aggregation. If you're screening RNA, you can gain a similar insight with plots of transfection efficiency. By iteratively feeding these findings back into future experiments or implementing tools like DOE, or design of experiments, Sunscreen will rapidly guide you towards your winning RNA and LMP formulation. Sunscreen and Stunner really are a match made in heaven. Each can cope with 96 samples in a single automated input, allowing you to make and measure your formulations in a single day. You can rapidly pull all sorts of insights from the data, as I mentioned on the previous slide. And we can now take a look at a couple of those data points in more detail. So looking here at the graph on the left, comparing two different structural lipids, we can see that when we use DOPE, we are generally seeing a lower PDI than with DSPC. So that's a useful trend for us to use going forward with different formulations. And in the right-hand graph, there are some clear differences in particle size when switching between cationic lipids, so changing from DOTAP to DDAB. Typically, the size is larger with DDAB. However, that trend is reversed when paired with a different structural phospholipid. So the trends are not quite as simple as interpreting just two variables. You have to look at the formulation as a whole. We also see that size actually gets smaller when the PEG lipid is changed to DMG PEG 2000. Because of the automation capabilities of Sunscreen, you are able to complete experiments in around 95% less hands-on time than sitting at an alternative instrument running experiments one by one. The low volume capability of the 96 well plates and the precise aspiration with our automated instrumentation means that you consume 60% less reagent than is typical from a manual one by one formulation instrument. And our reusable microfluidics, our glass sunnies, mean that you use 100% less consumables with no single experiment costs. Additionally, the rate at which we can generate experiments with this level of automation leads to an ability to generate around 5 times as many samples in a given time frame than using alternative manual one-by-one processing techniques. I've mentioned Stunner a couple of times so far in the presentation, so before we move on to the rest of the Sunny Suite, let's take a look at Stunner in a little more detail. So Stunner helps you to nail down your nanoparticle quality by knocking particle concentration, total RNA, size, PDI, and detection of aggregates all off of your list in one shot. So you'll know super quickly if your nanoparticles meet the mark or fall short. Stunner is the only system that pulls together UV-Vis concentration and fit-for-purpose DLS on a single 2 microliter sample on a 96 well plate format. Just load up that 2 microliters sample into a Stunner plate. You don't need to bother with sample prep or dilution, as the microfluidic plate allows for coverage of a wide dynamic range of concentrations. UV-Vis will get you the total concentration of RNA in the sample, while the fit-for-purpose DLS means that you're going to get the right kind of DLS for your particular sample. And we'll cover that shortly. Here we can see exactly what sample loading looks like with the help of some fluorescein and a blacklight. In 2 wells, we've already added 2 microliters of fluorescein. As you can see, to load another sample, you simply add 2 microliters into that input well and the sample wicks into the plate. Now, small things like antibodies scatter light evenly and in all directions, so angle tends not to matter. And when doing DLS, one angle is fine for small things like antibodies and proteins. However, larger things like LNPs, the story about the DLS measurement angle gets more interesting. When LNPs are all the same size, and everything will look consistent across all the angles, much like for an antibody. But when aggregates are present, as represented by the graph on the right, light scatters differently at different angles. So forward angles tend to pick up on aggregates first, while the backscatter angles don't see them. Stunner allows you to track size across many angles and see if you have consistent size or whether there are aggregates mixed into your sample. The way Stunner does this is by taking a single DLS detector and rotating it through multiple angles. This gathers data typically at 7 angles to get enough information to handle larger particles properly. We call it rotating angle dynamic light scattering, or RADLS. And here's what it looks like as the detector rotates around those different angles. To prove the benefits of looking at different angles by RADLS, we're going to run a spike-in study. So we'll take a good LNP sample of about 80 nanometers in diameter, that's nice and homogeneous, and add in an aggregated LNP sample that's much larger and more heterogeneous. And we can add small volumes of that large aggregated sample into the good sample in increasing quantities and see what they look like when we measure by RADLS. To analyze the mixed samples, we're going to use 2 data points. The first is Z average at 0 degrees, which is the extrapolation of all of the DLS info that Stunner has to the 0 degree scattering angle and gives you one clear output that is angle independent, meaning you have the best ability to look at the average size of all your particles in solution and not miss aggregation because of the angle your DLS uses. We'll compare that to Z average at 0 degrees to the Z average that you would get from a backscatter angle. Those angles are typically nearest to 180 degrees, where the laser basically bounces back towards itself. Those angles are most likely to miss aggregation. So let's take a look at that spike-in study and see what the data shows. We're looking at those backscatter angles first. As we add more and more of the aggregated LNP into our sample, the average size by backscatter DLS doesn't actually see any change in size. Likewise for PDI, there's no real discernible difference between any of these samples, whether there was no aggregate spiked in or 5% of the sample is aggregated. However, if we look at the Z average at 0 degrees, we can see very clear trends as we add more and more of the aggregated LNP sample into our good LNP sample. We see a significant visible increase in the Z average at 0 degrees, and an increase from 78 nanometers all the way up to 244 nanometers that was completely missed by only looking at this sample with backscatter DLS angles. Now, when you're optimizing nanoparticles and formulations to try and select the best performance and samples that are the most suitable for clinical environments, it's really important to be able to tell the difference between a sample that has no aggregates and one that has a significant quantity of aggregates, which is why RADLS is so key and is so valuable to nanoparticle researchers. Stunner also has a very high dynamic range when it comes to measuring samples by UV vis. And you can see here data across a 3-log dynamic range showing really nice linearity as our particle concentration increases. So you don't need to worry about getting to a perfect range of concentration. Whatever batch of LNPs you're making, small or large, fast or slow, Stunner can get you the answers that you need on a stock sample so that you can quickly compare batches or formulations and decide on next steps and eliminate unnecessary and time-consuming dilution steps with a need for them to be extra precise. One of the reasons that typical cuvette-based DLS instruments don't also look at UV-Vis at the same time is that cloudy solutions of LMPs and other nanoparticles can prevent the measurement by UV-Vis. But with Stunner's short UV-Vis path lengths, we can get those answers. We can cut through the turbidity with the unmixing application and check out just the absorbance signal from your payload. It's really simple quantification and it frees you from complicated, disruptive workflows, costly dyes, and wasteful standard curves. And as mentioned, with Stunner's 96 well plate format, you can run 96 formulations and get DLS and UV-Vis data within 1 hour, or within 2 hours if you want to use the Raddles measurement. Here we can see 96 replicates of RNA LMPs, where the green is the RNA concentration curve, which is also stated as a concentration below each graph, and the grey is the turbidity signal that's been separated. The coefficient of variation for these data points is a spectacular 1.54% over all 96 formulations. We talked at the beginning of this presentation about how different researchers are publishing on the methods they're using to use LMP formulation to improve things like targeting by either playing with size or composition or actively using targeting ligands by functionalizing the surface of the particles, so using antibodies or antibody fragments. While characterizing LMPs on their own is already difficult, characterizing antibody LMP conjugates is even harder. How do you get an RNA concentration, antibody concentration, and their ratio, and all the rest without spending hours in the lab? Well, Stunner's unmix algorithm allows you to specify 2 absorbance spectra that you want to quantify. So here we're looking at an RNA LMP with IgG spiked in. By defining that this sample has RNA, IgG, and of course the turbidity and lipids from the LMPs, Stunner will deconvolute the spectra measured and the output of the RNA concentration, your protein concentration, and their ratio, alongside particle concentration, size, and PDI, all at the same time, on just a 2 microliter sample, making it so much easier to optimize your product and allow it to easily reach its target. So let's get back to the Sunny Suite. Once you've got some formulation hits identified, Sunshine steps in to help finesse these formulations by exploring the impact of process conditions such as the total flow rate ratio, inline dilution, mixer type, and concentrations, to eventually transition into an optimized continuous flow experiment. Sunshine can run a series of automated experiments just like Sunscreen. However, with Sunshine, you use the same reagent for each of those experiments, loaded into the instrument with a single injection step, and Sunshine will release your reagents into the system a little at a time to execute each set of experimental conditions. When you have a combination of formulation and process that's giving you the results you need, Sunshine also has a continuous mode, which allows you to turn on the tap and make as much of your formulation as you need. In addition to being the ideal tool to transition screening experiments into an optimized manufacturing process ready for GMP, Sunshine's also a fantastic exploratory tool for researchers investigating all sorts of nanoparticle types. The flexibility of the mixing conditions combined with the range of mixer types and high solvent compatibility makes Sunshine, and for that matter Sunscreen, just as well suited to exploring things like polymer nanoparticles or liposomes, for example. Here you can see what that process looks like with some data. A quick triplicate screen of total flow rate shows how the particle size responds to flow for a specific Sunny and a specific formulation, with high consistency here between each run of 4 experiments. And then a larger volume production run collected in aliquots shows the consistency of particles produced over time in Sunshine's continuous mode. When you've optimized a continuous flow process on Sunshine, transferring it into a GMP environment couldn't be easier. Sunbather uses the same pumping technology and sunnies as both sunscreen and sunshine, allowing you for direct process transfer without the need for extensive re-optimization and revalidation. Sunbather can run all the same process conditions as sunshine, but it's an instrument that's underpinned by all the necessary documentation that you'd expect from a GMP instrument with fully replaceable wetted flow path that you can also reuse. And as you can see from the data here, SunnySuite instruments produce the same consistent particles across the suite at the same process conditions. So here you can see DOTAP LMPs that have been formed on each instrument and on Sunshine in both protocol and continuous mode, showing high levels of particle size consistency and low PDI. So let's take a look at how some of our instrument users have been putting them into practice. Nanovation UK showed how straightforward it was to match their existing particle formation process to sunscreen across a range of different formulations, including some commonly known ones and some of their own proprietary formulations. The results they found are consistent with common alternative processing methods, but with the advantage of being fully automated, less reagent hungry, and faster to run. So overall significant gains in efficiency whilst being able to get the same high quality results. A more detailed characterization of the particles also confirmed that the structures of particles by cryo-TEM were consistent with their expectations, and the ex vivo and in vivo data showed consistency with standard methods. Over at the Karolinska Institute, DNA LNP formulations are being screened for the purpose of targeting glioblastoma cells in the brain. Sunscreen enabled the exploration of a range of different formulations rapidly, allowing the team there to hone in on the most promising results quickly. The table on the right shows transfection efficiency for a range of different formulations processed by Sunscreen, enabling the most promising formulations to be quickly identified. In the videos, we can see time-lapse fluorescence microscopy data for 2 of these promising formulations. In the first video, we can see the cross-section of an organoid imaged overnight, showing the localized cell transfection in the neurons. And in the second video, we can see one of those fluorescing neurons actually dividing. Lastly, over at Early, the team are using Sunscreen, Sunshine and Stunner to boost their workflow for early stage cancer diagnostics and therapy using their targeting platform. Similar to the Karolinska Institute, the team at Early were able to screen a much larger number of formulations than was previously feasible to highlight the most effective formulations for further testing. By collecting data on both expression and uptake, a map can be created showing how different compositions behave so that trends can be highlighted for further development. The low volume consumption of sunscreen was also key when using dye-labeled DNA and lipids, which are particularly costly to work with when volumes are large. By using Sunscreen and Sunshine together, the team at Early are able to quickly scale promising formulations without optimization, as and when needed for animal studies. In summary, the SunnySuite instruments enable a seamless workflow for nucleic acid containing LNP drug development by using the same pumping and mixing technology every step of the way. Whether you're screening hundreds of nucleic acid payloads and lipid formulations to find a winner, optimizing a process to nail down perfect particle performance, or transitioning that process into the world of GMP manufacturing, the SunnySuite has you covered. And for characterization, Stunner gives detailed insights on both the payload and the particle using its unique combination of UV-Vis and DLS on 96 2 microliter samples in a single run to help you hone in on perfect therapeutic nanoparticles. Thanks for listening, and I'd be pleased to take any questions you may have.
안녕하세요. 자연과학의 김보영이라고 합니다. Q&A 시간을 가지기 전에 간단하게 언체인드 랩사에 대해서 소개해드리고 Q&A 시간을 가지도록 하겠습니다. 언체인드 랩사는 미국에 본사를 두고 있는 회사로 바이오로직스 및 유전자 치료제 기반 백신 개발과 관련된 다양한 연구 장비를 공급하고 있는 회사입니다. 바이오로직스 관련돼서는 이 전체적으로 언체인드 랩사 장비는 스몰 볼륨으로 좀 더 빠르고 쉽게 기존 방법보다 빠르고 쉽게 측정할 수 있는 다양한 장비들을 공급하고 있는데요. 2람다의 적은 볼륨으로 한 번에 96개의 핵산과 단백질을 정량할 수 있는 루나틱이라고 하는 장비를 공급하고 있고, 35람다의 적은 볼륨으로 한 번에 10개의 샘플을 1분 내에 측정할 수 있는 허니번, 그리고 8람다의 적은 볼륨으로 한 번에 96개의 샘플에 단백질 제재나 제형의 안정성 스크리닝 할 수 있는 안티라고 하는 장비를 공급하고 있습니다. 또 유전자 치료제 관련된 AAV나 LNP 어플리케이션 관련돼서 앞서 설명드렸던 스터너라고 하는 장비, 2람다의 적은 볼륨으로 한 번에 96개의 샘플을 정량하고 DLS를 측정할 수 있는 스터너라고 하는 장비를 공급하고 있고, 소개해드렸던 저희 써니 스위트, 선샤인, 선스크린, 선베터라고 하는 장비를 공급하고 있고요. 그리고 버퍼 익스체인지 관련돼서 버퍼 익스체인지 컨센트레이션을 자동화할 수 있는 운하기 빅투나라고 하는 장비를 공급하고 있는 회사입니다. 그리고 엑소전 관련돼서는 레플리콘, 아마도 엑소뷰라고 하는 이름을 한번 들어보셨을 텐데 엑소조 캐릭터라이제이션 관련된 장비를 공급하고 있습니다.